This is question 5 of the paper 2 2017 A-level paper. It's actually really one of those irritating questions for students because it doesn't ask them about what they know or about what they've read. It demands those skills that take time to develop, like evaluate, synthesize, suggest. And because of that, in answering the question, it doesn't leave you with any sense of satisfaction. Let's have a read through and see how we go. Arbuscular mycorrhizae fungi, AMF, are fungi which grow on and into the roots of plants. AMF can increase the uptake of inorganic ions such as phosphate. Suggest one way in which an increase in the uptake of phosphate could increase plant growth. So before we go to the question, just pause the video and have a think what you would write. Well, if you think about it, there are lots of things that phosphates are used for: to produce ATP or ADP, phospholipids for cell membranes, DNA or RNA, ribulose biphosphate, triose phosphate, and so on. The next part of the question asks, suggest one way in which AMF may benefit from their association with plants. So we know how the plant benefits from association with the fungus. How does the fungus benefit from association with the plant? So the fungi can now obtain carbohydrates from the plants, such as sugars. Remembering that it's only plants that can utilise light energy and turn it into chemical energy in the process of photosynthesis to make carbohydrates from carbon dioxide and water. So the fungi are able to take that from the plants. It's too vague to say that the fungi would benefit from the product of photosynthesis. And that's because oxygen is also a product of photosynthesis, but the fungus doesn't need to get oxygen from the plant. It can get it from the air itself. So be specific, be explicit in what you mean to say. The next bit of the question involves quite a bit of reading. I would suggest as you read through it, you chunk it up, you use your pen to break it up so that you can skip back to it and refer to the main points more easily. Scientists investigated the effect of different AMF species on the productivity of the plant community of a prairie grassland ecosystem when growing in or on soil containing different phosphate concentrations. The scientists set up identical plots of prairie grassland soil containing seeds of plant species found in the ecosystem. The scientists added different AMF species and different concentrations of phosphate to particular plots. Control plots without AMF species were also set up. After 20 weeks, the scientists determined the shoot biomass for each plot, and the results the scientists obtained are shown in figure 5. And I think the only bit of information there that isn't contained in the graph is that it took 20 weeks for them to determine the shoot biomass. So let's have a look at the actual question. Explain why an increase in shoot biomass can be taken as a measurement of net primary productivity. Now, measuring dry biomass is the only accurate way to truly measure growth. When we're dealing with large plot sizes like this, we can certainly use an increase in shoot biomass as an indication of an increase in dry biomass. And by measuring the shoot biomass, we've already taken out the respiratory losses of that plant. So the shoot biomass already represents the gross productivity minus the respiratory losses, which leaves us with our net productivity of the plant. Scientifically, we would then dry that out and have the dry biomass, but certainly the shoot biomass on its own is some way to represent that. The next part of the question is really quite awful. The best thing about it is to remember that there are actually 7 points that you could get according to the mark scheme. It says, using data from figure 5, evaluate the effect on plant productivity of adding AMF species and adding phosphate to the soil. So I'm asked to compare two things, and there's only 4 marks. So I'm going to compare the first set of 4 bar charts against the next set of 4 bar charts first of all. In other words, I'm going to compare the effect of phosphate. And as I compare normal soil phosphate with twice the soil phosphate, I'm going to scroll on my graph because I don't want to lose my way, I don't want to repeat myself, and I don't want to lose track of what I've already mentioned. So the first thing I've noticed is that for the control group, if I grow it in more phosphate, I get greater growth. Brilliant, that's 1 mark in the bag. The second thing, I could talk about Scuttlespora fulgida. However, it doesn't look very interesting, not much of a difference. So what I'm going to do, I'm going to talk about Entrophospora infrequens, which actually decreases when I put it in more phosphate concentration. Equally, I could talk about Glomus chlorodium. However, I've made 2 points about the phosphate concentration, so I'm now going to go on and talking about the AMF species instead. And so what I'm doing now is I'm just looking at the first 4 bars on that chart, and I'm comparing them against the first bar, which is the control. So the first thing I'm going to say is that Scuttlespora fulgida reduces growth, whereas Enterospora infrequens and Glomus chlorodium both increase growth. And that's because I'm just looking at that 4 sets of bars, comparing it to the first bar, which is the control. I double check back to the stem of the question. It's asked me to use the data. So I've done that. I've talked about increases or decreases. And then it asks me to evaluate. So then I need to move on to the next part, and I need to look at the study that they've done. Well, the first thing is there's no statistical test. So although I've talked about it increasing or decreasing, I don't know if that's significant or not. So that's the first point I'd make, that there was no statistical test done to determine if the differences were significant or not. The other thing is that it was only tested over 20 weeks. Is that long enough? Also, we only measured the shoot biomass. We didn't measure the biomass of the whole plant. They might have had extensive root systems. So we don't know what growth has taken place underground. So you just needed to get 4 marking points out of a possible 7. There were lots of things you could say, but how do we tackle it? First of all, we looked at comparing phosphate and non-phosphate, so the first 4 bars against the next 4 bars, and then we looked at the AMF species, so we were just looking at the first 4 bars, comparing the latter 3 of those 4 against the first. And we're going to use a pen to deface that diagram because we don't need it after this question, but we certainly need to make sure that we don't repeat ourselves or lose track of what we're trying to say. And the next question says, using the e to the x button on your calculator, determine the rate of shoot biomass production in grams per day for the control plot in soil with normal phosphate concentration. So you don't need to understand the maths of this. All you need to know is that e to the x is the opposite of log to the e. And you just need to know how to punch that into your calculator. So when I read off my graph, I get roughly about 2.4. So I'm going to stick that in the calculator, e to the 2.4, and then it gives me my answer, but I need to know it per day. That was for the whole 20 weeks. Well, clearly 20 times 7 is 140. So I'm then going to get that number and I'm going to divide it by 140. And that gives me my answer if I round up to 0.08. And according to the mark scheme, a range was accepted there, anything from 0.07 to 0.09. So there we have it, a biology skills-based question where all the revision in the world wouldn't help you with that. The only thing that would help you with that is by having a go at past paper questions and answering them not just with the mark scheme, but in conjunction with the examiner's report. Of course, that's time consuming, so that's why I've made these videos, so hopefully it'll save you a little bit of time. As always, I hope you found it useful.